Good afternoon, everyone. Am I audible to you all?
Yes, ma'am.
Okay. I'll share my screen. So, is the PPT visible?
Yes, ma'am.
Okay, thank you. So, we are in the week 4 of the Proteomics course, and we will be discussing today problem solving session for the week 4 content. And so first we will start with few questions that some of you had in the last session regarding assignment 1 and 2. So there were, I guess, one question from assignment 1 and two questions from assignment 2. So we will discuss that first, and then we'll solve few questions related to week 4 content. So coming to so this question is from assignment 1. Just a minute, I have to admit some people. Okay. So this is the first question. This question is from assignment 1. The last question, it is upon BLI technique. So basically, BLI technique we is used. It's an unlabeled, label-free technique to learn or to study the interaction between two proteins or protein like molecules. Okay, so this is the diagram of the BLI experiment. So someone has done a BLI experiment, and the binding affinity of three distinct molecules with ligand X was determined. So there are three distinct molecules A, B, and C, and ligand X is a ligand with which all the three biomolecules are interacting. So from the graph, we have to clarify that which of the following options is correct. Okay. So option 1 is biomolecule A has a higher affinity for the bound ligand X. So the principle of BLI technique is that the shift, so you can see higher the shift, higher is the affinity of a particular biomolecule to the ligand. Okay, so it is clearly visible from here that A is having lesser shift compared to B and then compared to C. So the molecule C is showing highest shift compared to A and B, and the shift in this experiment is directly proportional to the affinity as well as to the density and the molecular weight of the protein. So more the protein, more the affinity, more will be the shift. So according to that, the C has the highest affinity. Okay, from the diagram we can see that. But here what is mentioned in the option 1, biomolecule A has higher affinity for the bound ligand X. So this statement does not hold true because the correct option is C. C has the highest binding affinity. Now option 2 is biomolecule C has the lowest affinity among others as it is showing the maximum shift. Okay. So this is opposite. The statement is completely opposite of what it is. So the maximum shift, if a particular biomolecule is showing a maximum shift, it means that it has the highest affinity and not the lowest affinity. So even this option is incorrect. Coming to option 3, amount of biomolecule B bound to the membrane is in moderate. So we can see here that the shift in case of biomolecule B is in between molecule A and C. So this holds true that the amount of biomolecule B bound to the membrane is in moderate. Whereas coming to option 4, all the three biomolecules have the same affinity and density. So if that was the case, then you would have not seen three distinct uh, shift for for the three biomolecules. So this statement is incorrect. So our correct option is option 3. Amount of biomolecule B bound to the membrane is in moderate. So anyone has any doubt regarding this? So let me know. Or is it clear now? Someone was having this query in the last session. Someone has raised the query. Is this clear now? The question is clear now?
Yes, ma'am. Clear.
Okay, okay, fine. So we'll move to the another question. So this is from the assignment 2. So this is regarding Bradford assay. So Bradford assay is used for quantification of the proteins, and the same kind of problem we have solved in our week 2 Saturday session also. So this is similar to that only, but even then I have discussed this. So let's just start. Sujata used the Bradford test to quantify protein levels in four sample A, B, C, D. She prepared the standards for the standard curve with BSA. The tables below show the absorbance average from her experiment. Okay, so this are the absorbance average of BSA. So just a minute. Okay, so this table shows the absorbance average of BSA at different amounts. Okay, and this is the standard curve prepared from these values. And yes, so also the absorbance value of the sample A, B, C, and D are given. So you can see these are the absorbance value of the sample A, B, C, and D. So now you have to find out if Sujata wants 35 microgram of sample D for another experiment, how much volume she should be using. So for a Bradford assay, generally 2 microliter of protein we are using. Okay, so depending upon that, first you need to calculate what is the amount of protein D. Okay, and from that, after that we can calculate that if we want 35 microgram of protein, then what is the volume required for it. So using this equation given, first try to calculate the value of amount of protein D. Okay. So everyone just try this and then we'll discuss. So you have to use this equation y equals to 0.0272x plus 0.0082, and using this you have to calculate the value of x for protein D, and y is the OD, and for sample D the OD is 0.308. So if anyone has calculated what is the amount of protein D, let me know what is that amount. This is the complete question. Has anyone calculated what is the amount of protein D? No one has yet done that.
Ma'am, I was asking ki for to calculate like x for will be 10308, then we have to find out y.
Yeah, your voice is breaking. Can you repeat the query?
I was asking ki for calculating the concentration, we need to put the value of x, which is uh, 0.308, and then we need to find the value of y.
No. So you have to calculate the value of x, and 0.308 is the value of y. You see here the y on y-axis the OD is given, and 0.308 is the OD that you have to take as y, and x is the this uh, x-axis, which will give you BSA amount. So here by mistakenly concentration is written. So you can see here in this uh, table, it's the amount of BSA. So from that you can calculate the value of X, which will be the amount of protein D. Okay.
It's 11.02.
Yes, correct. You're right. Now, 11.02 microgram, and you are using 2 microliter of sample for your Bradford assay. Okay. So, using that information, now can you calculate if the what if your amount is 35 microgram, what will be the volume required? So generally for Bradford 2 microliter of your sample is required. Okay.
No, are you saying something, ma'am?
Sorry.
Are you saying something?
No. Have you calculated for 35 microgram?
No, I cannot understand that part.
Okay, so the thing is that for Bradford assay, generally you are using 2 microliter of your protein sample. Okay, whenever you are performing Bradford test. So, whatever amount you got for protein D, so someone has answered 11.02 microgram, which is correct. So, that much amount is present in your 2 microliter of sample because you are using 2 microliter of your protein sample for your Bradford assay. Okay. So now, if you have 11 micrograms of protein, uh, when you are using 2 microliter of your sample, then what will be the volume if you are using 35 microgram? Because the question is you require 35 microgram. So what will be the volume? So it's a simple multiplication, cross multiplication that you have to do. Okay, and so I'll just show you. So first of all, you need to calculate the amount of protein D. Okay, so using this equation, you can calculate this value and you will get 11.02 microgram, which is the amount of protein D. Now, for Bradford assay, generally you are using 2 microgram of sample, sorry, 2 microliter of sample for your assay. Okay, so when you are using 2 microliter of sample, this is the amount of your protein present in the sample. Okay, so if you want 35 micrograms of of your protein sample, then what will be the volume? So you just have to, so this is just a cross multiplication, 35 into 2 divided by 11.02, which is 6.35 microliter. Okay, so this is the volume of your sample that you have to use if you want to, to load, or you want to use your protein sample, provided that this is the amount. If you want 35 microgram of protein in your sample and you want to use it, then you have to use 6.35 microliter. So is it clear now? So this is same as we had discussed in our session 2, our Saturday session 2. So there we have discussed the similar kind of problem for Bradford assay, and we had also calculated for all the four different proteins. Okay, so this is the same kind of question. So is it clear now, or anyone is having still any confusion? Then let me know.
Ma'am,
Yes.
where the volume is not mentioned, we have to assume that it is always 2 microliter.
Yes. I agree that volume is not mentioned, so it should be mentioned. Um and calculation is based upon 2 microliter. So,
Yes, ma'am. When we got the answer, then back calculation showed that it should be 2 microliter. But we have doubt that
okay.
previously the volume was not mentioned.
I'll ask another TA if she can comment anything upon this. So, is Medha or Shashwati here? If they can comment upon this, like, do we have to use 2 microliter always, or it should be mentioned?
Hello everyone, I'm Medha. I'm not sure about Shashwati. Um, right now, actually, sorry for any background noise if if it is there. Um, so usually in the question it will be given how much sample has been taken for to do the quantification. But we do it in our lab very regularly, and we usually use 2 microliter because sample taken less than that will give you a lot of error. So we usually prefer taking at least 2 microliter, but depends on the user also. Sometimes the user might use 10 microliter or so. So most of the time it is specified in the question. Particularly for our course, if it is not specified, just consider it 2 microliter. But otherwise, in daily life, when you are applying it, just keep in mind how much sample you have used while doing the quantification.
Okay, ma'am. Thank you.
Okay, so now we'll move to another question. So this is another question from our assignment 2. So Arya has performed protein extraction using trizol method and has obtained 720 micrograms of protein from her sample. She has to perform two-dimensional gel electrophoresis by using 18 centimeter strip and wants to identify her protein of interest. further using mass spectrometry. Select the appropriate amount of protein she should be using to start her 2DE experiment. So, this question is basically dependent upon what amount. Just a minute. Okay. Yeah. So here it's based upon the concept that what is the amount of protein required whenever you are performing 2DE experiment. Okay. So so the correct option is the option one, 300 to 100 microgram. So now if we see option 2 and 4, so comparatively they are lesser than option 1, and it will not be sufficient enough whenever you want to perform 2D electrophoresis, especially when you are using 18 centimeters strip. Now, option 3 is 1000 to 1500 microgram of protein. Now, the protein that you have obtained is only 720, so this cannot be the correct option. So basically, this is somewhere much closer to what is generally um, generally practiced. So the correct option is option 1, because option 2 and 4 will be insufficient compared to option 1. So this is the more closer to what is required in the laboratory scenario. Okay, so
No,
now, yes.
no, it is like standardized. Like for this sample, we need to use such a
It's not
standard.
standardized as such. It's generally there are ranges with which you have to work. So, as in the Bradford essay, as Medha mentioned that uh, generally 2 microliter they are using in their laboratory, right? So it's not that with 10 microliter you cannot perform the assay, or with 1 microliter you cannot perform the assay, but maybe for the kind of proteins that they are uh, using, or for the assay that they have standardized in their lab, the 2 microliter is working as a it's giving an most appropriate result. So in a similar way, it's not that you cannot use this, but you will not get a appropriate result as you are supposed to get. So higher amount of protein, it may lead to giving you a bigger bands, and you might not be able to see distinct bands. And if the you are using low amount of protein, then you might not be it won't be able to separate out. You might miss out on some very low abundant proteins. So that is why generally a range is given, a standardized there is a range, there is not a fixed one number. And then depending upon your experiment, you have to figure out which amount, or which number is standardized for your particular experiment. Okay. So 100 to 300 is like uh, n- a general term, a general microgram of protein used for a 2DE experiment. So this will give you a better result compared to higher amount or lower amount of protein. Okay, so shall we move to another question now?
Yes,
Okay. So now,
Yeah, Sneha,
yes.
just to add to what you said, so in the previous question, so here they have mentioned that it is an 18 centimeter IS strip that we will that the experiment is using. So depending on the length of the strip, also how much sample you load varies. So for a smaller strip like 7 centimeter or 13 centimeter, depending on the size, there are different sizes. So the amount of protein, total protein that you'll be loading will be less. 18 centimeter strip, you can imagine then the gel will also be bigger, right? Because this will be the length of the gel as like 18 centimeters. So at least 18 centimeter the gel will be. So then the amount of protein loaded should be more so that the spots are properly visible, the protein spots. But it should not be too much so that there is no resolution between the spots. So that's why there is, as Sneha said, there is a particular range that you work with.
Okay, thank you, Medha. So let's move now to another question. So these are a few questions from the week 4 content. So this is the first question. Which of the following approaches is not used for proteomic analysis in systems biology studies? So mass spectrometry, microarrays, 2D page, and say so. Unfortunately, I forgot to remove the correct option. So SAGE is basically used for transcriptomic analysis. So the full form is okay, I forgot that. It's a genomic expression. Uh, so basically, it is used to study the transcriptomic levels. Whereas if you see 2D page, mass spectrometry, and microarrays, all of these are used for proteomic studies. So this is an another question. Okay. So Aryan performed dyes labeling for 3 samples for which he mixed appropriate amount of protein with that of respective labels. He kept the samples as such for half an hour. After that, he added more of lysine solution to the sample. What do you think would be the purpose of adding additional lysine? So option A is this step is required to avoid any unspecific labeling. This step is required because skipping this step might lead to unexpected increased pot volumes. If this lysine addition was not done, he would not have got any of the protein labeled. Option D is this step was not required, he spoiled his experiment. So, what is the correct option, or what are the correct options in the given 4 options? So, can I get some response? So, what is the purpose of using lysine in dyes experiment?
Is it A and B?
It's A and B. This step is required to avoid any unspecific labeling, correct? This step is required because skipping this step might lead to unexpected increase pot volumes. Correct. So you are correct that lysine helps in avoiding any unspecific labeling during your diets experiment. So you can also refer to I guess second video of week 4 for this. So let us move to another question. So, okay, I have forgotten to remove the options here. So, so here are uh, 3 images given. Given below are the few gel images for single proteomic experiment. Which of the following approaches would have been used to obtain such gel images? So, 2DE gel stain with fast green, Nile red stain, and Kumasi stain. Option 2 is stain with fast green, FCF, Pro Q diamond, and Kumasi stain. Okay, I think someone wants to join. Just a minute. So just a minute. I'll stop sharing. Uh, I'll share it again. Just a minute. Okay, so is the screen visible?
Yes, ma'am.
Okay. So, unfortunately, I had forgotten to remove the correct options. So, basically, you can see there are three different images from a single proteomic experiment. So, you would have seen that the correct option is option 4, dyes label samples followed by 2D experiment and scanning at different wavelengths. So, generally, here Psi2, Psi3, and Psi5 dyes are used for the dyes labeling, and all these three dyes has different excitation and emission wavelength, and that is why at at different wavelengths you are uh, seeing your gel, and you are getting three different images from your single proteomic experiment. So, and because your sample is labeled with three different kinds of dye, you are getting three different images. And this is the approach we are using for dyes. We are using Psi2, Psi3, and Psi5 dyes. Okay. So the correct option is option 4, dyes label samples followed by 2D experiment and scanning at different wavelengths. So this is an another question. Which of the following statements is not correct about side dyes? So side dyes are used for this dyes experiment, as I discussed in the earlier question. So side eyes are photostable in nature, side eyes are pH insensitive, side eyes are isobaric, and side eyes are spectrally distinct. So you have to select the option which is not correct about the side eyes. So anyone wants to comment upon that? Which of the following option is not correct about the side eye?
It's pH insensitive.
It's pH insensitive. Okay. Uh, any other response? Uh, any other one wants to guess?
No, option C.
Option C. Side eyes are isobaric. Okay. So the correct option is C. Side eyes are isobaric. So if you have gone through the lecture 2 of the week 4 content, so side eyes are photostable, they are also pH insensitive, and they are spectrally distinct. Okay, but they are not isobaric. So the correct option is option C. Side eyes are not isobaric. Now coming to another question. So Sandipan has a set of samples including two different conditions labeled as condition A, excuse me, and condition B respectively. He planned for a dyes experiment for his samples in which the samples from condition A were labeled with Psi5 and those of condition B were labeled with Psi3. Okay. So this is an important information. Samples A were labeled with Psi5 and sample B were labeled with Psi3. For internal standard, they are using Psi2. So individually labeled sample as well as different pulls of them were taken for SDS page. Following it, the gel was scanned to visualize the protein bands. Given below is the representation of merged image of gel obtained after scanning at the required wavelengths. So basically this image if is of a sample at condition A, sample at condition B, then he had also pulled the samples together, and and the bands are also for that. So you have to identify, depending upon the image given, which of the following is the correct match of the gel lanes 1 to 5 with respective types of labeled sample runs. So you have to identify which is the which of the this is for sci-fi, or which is for sci-3, or whether it is a mixture of sci-fi, sci-2 and sci-3, or only a combination of two dyes. So which is the correct option of the four? So think about it. Uh, See the image, see what is already mentioned. Condition A and condition B. So condition A were labeled with psi 5, condition B was labeled with psi 3. So now what is the correct answer? Think from the image and let me know. I'll give you some time. Any guesses? Anyone wants to guess?
Options
Option, which option?
C.
Option C. Okay. Any other guess? Anyone else who wants to try?
Some people in the chat are saying it's option D.
Okay, option D. Okay, so the correct option is C. So if you see lane 1, so sci-fi is generally red labeled, uh, green is psi 3, and blue is psi 2. So if we see the lane, so 1 should be psi 5, 2 should be psi 3. So in the option D, 1 is psi 2. So this is incorrect. Psi 2 is generally for blue. So here 1 is psi 5. So option B and C has 1 is psi 5, which is correct. Option 2 is psi 3, which is correct for B, C as well as D option. Now blue is for this, which is lane 4. So first we will see these three and then the other two lanes. So for 4 it should be psi 2. So that is mentioned in option C, option D as well. Okay, yes, option D as well. Option B, it is different. So in option D, the first is incorrect. So in only in option C, uh, lane 1, 2, and 4 are correct, and then coming to 3 and 5. So the pulled sample of Psi5 and Psi3, it will give yellow colored pigment. And whenever you are pulling all your condition A, B as well as the internal standard, so generally Psi2 is used as an internal standard for dyes experiment. Then when you are mixing all the three, you will get this band. So your correct option is option C. Now, coming to this is a follow-up question for the previous question. So, yellow and white colored bands in the given image represent which of the following? So, 3 and 5. These two lanes represent which of the following? So, yellow bands represent the protein present in samples of condition A only, whereas white bands are for those protein present in samples of condition B. Okay, so yellow band represents protein present in sample of condition A only, which is not true because if, if, even in this answer we have figured out that fifth, the yellow band is from Psi5 and Psi3 dye. So protein A, protein A is Psi5 dye, so it also contains Psi3. So the option 1 is incorrect. Coming to option 2, yellow bands represent the proteins present in sample of condition B only. So even that is not true because yellow band represent a combination of Psi3 and Psi5 dye. So option B is also incorrect. Now coming to option C, both the colored bands represent protein present in both the samples of condition A and B. So, what do you think? Is this the correct option? Can this be the option? Can this be a correct option? Can I get some response?
Yes, ma'am.
So
In the
do you,
previous.
right? So both the colored bands represent proteins present in both the samples of A and B. So as we have already discussed in our uh, uh, previous question, here that 3 and 5 white and yellow band are a combination of psi 5 3 psi 5 psi 3 psi 2 for white and psi 5 and psi 3 for yellow band. So and you are labeling your both A and B condition with psi 5 and psi 3 dye, and you're using psi 2 as an internal standard. So which means that both the colored bands have the proteins from samples of condition both A and B. Now coming to option D, both the colored bands represent protein present in samples of condition A. So this cannot be correct because here uh, we can see from this option only that yellow band is from psi 5 plus psi 3, which is uh, condition A and condition B. You are labeling condition A with psi-pi and B with psi-3. So this band also results from both the proteins from both the condition. And here along with the condition A and B, you are using internal standard as psi2. And internal standard is nothing but a pooled sample of your condition A and B sample. So that is why your option D is also incorrect. Your correct option is option C. Both the colored bands represent proteins present in both the samples of condition A and B. So coming to the last question of this session. For labeling the proteins, most of the time the amino acid lysine is targeted. What could be the most possible reason for targeting this amino acid? Option A, it is positively charged amino acid at the physiological pH. There is a very high probability that every protein has at least one lysine residue. It contains primary amine group. It is an essential amino acid. So what is the correct option?
B, ma'am.
There is a very high probability that every protein has at least one lysine residue. Correct. So this is the correct option. That is why you are using lysine when for your labeling experiment. So lysine is a minimal labeling, and there is another kind of labeling also, cysteine labeling, which is saturating labeling, and it is more sensitive, but uh, lysine labeling is most preferred kind of labeling for DIGE experiment. Okay, so this comes to the end of today's session. So even if you have any doubts, you can ask now and let me know.
Am I audible?
Yes, you can go ahead.
Ma'am, I have a question.
Yes.
In week 3, I could not submit week 3 assignment. Is it hamper my certification?
uh, your voice was breaking. Week 3 assignment, what is the problem with week 3 assignment?
Ma'am, I could not submit it.
Okay, you cannot submit it.
So, is it hamper my certification, or am I eligible for the examination?
Uh, I think Medha, you can comment upon this. She had not
Yes, so
submitted.
usually, I can confirm this, but usually I think it's an 8 week course, right? So one or two assignments they is okay. Like if you miss, you have done zero, na? Assignment zero is done.
I
One,
have submitted week 1 and week 2.
three you could not know. So if you attempt all other assignments like without missing, then it won't be an issue.
Okay, thank you.
Okay, so any other doubts? Okay, then I'll stop sharing and we can end the session and see you for the next session. Bye bye.